first edition of the BIO-CAT Science Education Series. Today I am here with Dr. Sean Garvey, who is the Director of Research and Development for BIO-CAT Enzymes. Thanks, Sean, for being here.
Hello, good morning.
Good morning. You have a crack team of scientists, including fellow PhDs. Can you elaborate what the team is focused on right now?
Sure. At BIO-CAT, we are exclusively focused on enzyme research and development. We're focused on enzymes for human health, animal nutrition, food processing, and ingredient upcycling. In human health, we're focused on using microbial enzymes to improve food digestion, to improve nutrient release and absorption, as well as to reduce gastrointestinal symptoms associated with food intolerances. In our laboratory here in Virginia, we test enzymes in our in vitro gastrointestinal digestion simulation to better understand how enzymes work and understand dosing for clinical trials. So in the lab here, we're asking, how do enzymes fundamentally digest macronutrients? How do enzymes interact with complex food and mixed meal matrices? We take those hypotheses and results from the lab and we test those in clinical trials with expert partners at universities and clinical research organizations. Demonstrating benefit in human participants is absolutely critical for credible, differentiated, meaningful enzyme product development.
What technology is BIO-CAT currently employing to elevate outcomes?
In research and even medical practice, oftentimes we're only as good as what we can measure. At BIO-CAT, we identified a need to better understand how enzymes transform dietary macronutrients. Our laboratory here in Virginia has been outfitted for decades with several high-performance liquid chromatography instruments, or HPLCs. And we can use these HPLCs really well to measure the final outputs of digestion. For example, the free amino acids that come from the digestion of proteins, the free fatty acids that are released from dietary fat digestion, and those free sugars like glucose and fructose that are released from digestion of carbohydrates like FODMAPs and starch. We needed to better understand the intermediates of macronutrient digestion. So 2 years ago, we purchased a liquid chromatography mass spectrometry instrument, or an LCMS. And with this LCMS, we were able to better understand how microbial proteases actually digest these canonically digestion-resistant peptides, like peptides from wheat gluten proteins and peptides from cow milk casein proteins. With the LCMS, we were also able to better show how an enzyme called fructan hydrolase, or inulinase, was able to transform these long-chain fructan molecules, a type of FODMAP that we find in inulin and garlic and onions, how inulinase can transform those fructans into free fructose that can be absorbed. So we've always known that microbial enzymes work to improve digestion, and with the LCMS, we're able to better know how.
Wow. Thank you, Sean, for answering all of our questions. Your knowledge is invaluable